Today we're going to be listening to two contestants debate about who is better than the other. Aubrey, will you please introduce our two contestants?
Of course. First up, we have DNA.
Hi, I'm deoxyribonucleic acid, but you can call me DNA for short.
Next up, we have RNA.
What's up? I'm ribonucleic acid, but you can call me nonchalant RNA. I can't wait to absolutely demolish DNA in this debate.
Yeah, right, as if you ever could.
Whoa, everyone. Let's calm down a bit. Nonchalant RNA, is it okay if we just call you RNA without the whole, nonchalant part?
Fine, whatever.
Sophie, what will they be debating about?
I think they'll debate about who's more important based on what they do for an organism.
I agree. Let's have them tell us what they are made up of first so we can learn some information about them. DNA, why don't you start us off?
Don't mind if I do.
What? Why does DNA get to go first? Why can't it be me? I'm obviously better.
We're just going in order. Don't worry about it. You'll be next.
Fine.
Well then, shall I continue?
Oh yes, of course. Go ahead.
I'm made up of three components: deoxyribose, which is a 5-carbon sugar, a phosphate group, and nitrogenous bases that form the building blocks of DNA.
Whatever, what does that even do for the organism?
I was just getting to that part if you would be patient. First, let me tell you, my shape is a double helix, and you can remember that by thinking of it as a twisted ladder. Second, there's a type of bond that holds the sugar and phosphate backbone of DNA together, and that is a covalent bond. Do you know what two parts make up the sugar-phosphate backbone?
No. What is it?
Deoxyribose and a phosphate group. Third, there are 4 nucleotides, nitrogen, bases that you need to know before I tell you what I do for an organism. And they are adenine, thymine, guanine, and last but not least, cytosine. You also need to know what they pair to. Adenine always pairs with thymine except when it's paired with uracil, which is part of RNA, so I'll leave it at that. And guanine always pairs with cytosine. Lastly, what I do for the organism is I store and carry genetic instructions for development, functioning, growth, and reproduction. Let me explain in more detail. First, DNA, aka me, has to replicate itself, and this usually happens during interphase.
I forgot what was replication.
Replication ensures that each resulting cell will have a complete set of DNA. The 3 main steps of DNA replication are initiation, elongation, and termination. During initiation, enzymes like helicase unwind the double helix at specific origins. In elongation, new DNA strands are synthesized by DNA polymers, using the original strands as templates, and this process involves creating a leading strand continuously and lagging strand in segments called Okazaki fragments. Finally, termination occurs when the DNA strands are fully synthesized and any RNA primers are removed and replaced.
Oh, I see. Okay, thank you.
Long story short, I serve as a blueprint for life, guiding the production of proteins that build and maintain the body. And I pass hereditary information from one generation to the next through replication. I think that is all about me.
Finally, why did it take so long for you to explain all that?
Because I wanted them to understand.
Okay, that's enough of this RNA. Go ahead and tell us about yourself.
Okay, so unlike DNA, I can travel outside the nucleus to get things done. I help in processes called protein synthesis, and I also help as a catalyst, a regulator of gene expression, and a component of some viruses.
RNA, may you explain in more detail because some of us don't even know what that means.
Oh, yeah, I guess. Let me see. I will start with protein synthesis, biological process of creating proteins from the instructions in DNA, which involves 2 main steps, transcription and translation. During transcription, a segment of DNA is copied into a messenger RNA molecule. Then, during translation, this mRNA molecule is used as a blueprint to assemble amino acids into a specific protein at a ribosome. If you remember what DNA said earlier about adenine and uracil? Well, adenine pairs with uracil in both transcription and translation, because uracil is the base that replaces thymine in RNA. During transcription, an RNA molecule is built complementary to a DNA template. So when DNA has an adenine, RNA will have a uracil. And when DNA has a thymine, RNA will have an adenine. During translation, the mRNA codons pair with tRNA anticodons. So adenine in the mRNA codon still pairs with uracil in the tRNA anticodon.
Wait, what does transcription mean?
It's basically just a coded complement of RNA.
Okay, and what do you mean by translation? and the mRNA, tRNA, rRNA, and stuff.
There are 3 types of RNA: messenger RNA, also mRNA, ribosomal RNA, rRNA, and last but not least, transfer RNA, tRNA. That is what I meant by those. And well, translation is when the mRNA message is decoded into a protein.
Oh, okay.
Where does transcription occur?
It occurs in the nucleus.
Okay, and don't mind me asking, but what is the role of mRNA and tRNA?
mRNA's role is to exit the nucleus and travel to the ribosomes to be quote-unquote read, and tRNA's role is to bring the correct anticodon and amino acid.
Okay.
Oh, and where does translation take place?
It occurs in the ribosomes in the cytoplasm.
And what is a codon that you keep saying?
A codon is a group of 3 nucleotides that codes for an amino acid.
Oh, and earlier you said that DNA can't leave the nucleus. Why not?
I can answer that one. I can't leave because I am the vital quote-unquote master plan and could be damaged. That is why RNA is here to leave the nucleus for me.
Oh, okay.
Yep, okay, let's get on with this. Next is RNA, aka me, can act as a catalyst through molecules called ribosomes, which are RNA molecules that fold into a 3-dimensional structure to perform specific biochemical reactions such as RNA splicing and peptide bond formation in the ribosome. I can also regulate gene expression through various mechanisms, including small regulatory RNAs like miRNAs and siRNAs that control translation, and mRNA stability, and long non-coding RNAs that can alter chromatin structure, transcription, and splicing. Lastly, I am a component of some viruses, the genetic material for some viruses, serving as the genome to store hereditary information. So basically what I do for an organism is I carry the genetic code, I build protein, I am the component of some viruses, and last but not least, I regulate gene activity. I'm obviously so much better than DNA.
What? No way. I'm better and more important.
I don't know. I think both of you are equally important for organisms in life.
I guess, but if you had to decide, then who would it be?
After a long time of thinking, Sophie and I have decided on who we think won the debate. Drum roll, please.
RNA!
Yep, you heard it. RNA is the winner. Bye, everyone.
Bye!